Welcome to the Biology 182 lecture series on classification. This video will discuss horizontal gene transfer. At the end of this video, students should be able to understand how divergence occurs horizontally between evolutionary lines, in addition to vertically within one lineage. Vertical transmission is what we know, and we have seen. This is where a parent gives its genetic traits to its offspring. And so this happens within a lineage. And you can see vertical transmission occur in the rise of synapomorphies within taxonomic groups, right? So the evolution of the jaw. All organisms in all of these lines pass on their heritable trait of the jaw. We know it's heritable because hagfish did not have a jaw, or lampreys that we talked about, they did not have a jaw. So jawless fishes, then arose jaws, and we can see all the organisms from that point have these jaws. Also on this other image to the right, you can see these are proteins and various components of our DNA that have changed and arisen and been passed on from parents to offspring throughout time as well. So vertical transmission is parent to offspring transmission. This is within a lineage, going vertically through the tree of a lineage. Horizontal gene transfer is DNA that is acquired from unrelated individuals. It's like me walking next door to my neighbor, putting out my arm and catching all their DNA, right? We are not related. Vertical gene transfer is the DNA inherited from a parental organism and passed on over time. Vertical transmission, you can see, this occurs in reproduction, right? Mitosis, meiosis, these are vertical gene transmissions. Even in cladograms, you can see this phylogenetic tree. C has a different gene transmission that it's giving from the original organism. And all of that line will continue to have that mutation or change, right? Vertical, however, crosses between organisms. So it's like C handed over some of this mutation that it had to B, and now B has it as well. This happens a ton in prokaryotes, right? We see it far less in eukaryotes, but a ton in prokaryotes. You can see here the new mutation arose in an organism, and now it's sharing it with its neighbor. It's like, here, have some of my new mutation that might help you in life, okay? So there are three mechanisms by which mutations are accumulated in horizontal gene transfer of prokaryotes. The first is conjugation, and this is where I'm a bacteria here, and I'm a bacteria here, and my bacteria share a sex pilus, and my DNA now is transferred to you. So now whatever I had in my DNA here with my plasmid, I'm giving to you. That is called conjugation or bacterial sex, right, through the sex pilus. Another way that bacteria or prokaryotes can ingest DNA is from the environment. They can just take it up. Sometimes it's like a bacteria is lysed and now there's a bunch of random fragments of bacterial chromosomes. And those fragments are picked up through endocytosis by another bacterium. Otherwise, it could be free floating from something else in the environment, may not just be a lysed bacterium. And they pick up this additional DNA and incorporate it into their genome. The third mechanism, aside from transformation, which is picking up from the environment, or conjugation, which is sharing with your neighbor, would be bacterial vectors. Viral vectors. I did not mean bacterial. Viral vectors. A virus injects its DNA, like a phage, right, injects its DNA into a bacterium. And this macrophage now had its DNA into the bacterium. And we know sometimes that bacterium incorporates it into its DNA and it goes into a lysogenic cycle where it lyses the bacteria to share the fragments of its DNA, right? That's one way that viruses transmit their viral DNA or RNA. It also could be incorporated into the genome and then be in a lysogenic cycle where it just replicates and then shares its DNA with its offspring. And additionally, through conjugation, it could pass it on as well. So the three mechanisms of prokaryotic horizontal gene transfer between species are through conjugation, or the sharing with your neighbor, transformation, or taking it up from the environment, or transduction, getting it from a virus. Transformation or transfer of genes horizontally occurs between domains as well. We see this with endosymbiosis, right? In that bacteria had plastids, and over time, we've seen plants have plastids. The mitochondrial form from sulfur or purple bacteria, right? The chloroplasts form from cyanobacteria, this engulfing of another organism and using and incorporating its DNA into your own, eventually over time becoming one organism. These are horizontal gene transfer that occurred millions and millions of years ago. So horizontal gene transfer occurs between domains. It also occurs within a domain. So even within a kingdom, this is between kingdoms of plant and fungi, right? We have fungi and plants that are intermingling their DNA laterally or horizontally, so that one loss of phagotrophy occurred in fungi and also occurred in plants in that homoplastic or homoplasy way. But also, if you look, things like the rise of the plant cell wall was shared between organisms that were plant and fungi. Granted, it diverged and took a different mechanism. Plant cell walls are cellulose, fungi cell walls are chitin, but they're laterally transferring or horizontally transferring DNA between organisms and between species. Eukaryotic horizontal gene transfer we see between bacterium and eukaryotes, viruses and eukaryotes, other eukaryotes, like we said, within plants and fungi, and then through endosymbionts, where we uptake other organisms and they become over time incorporated into our cells. You see it with mitochondrial DNA and nuclear DNA. The transfer between MT and N DNA actually has occurred. And all of these various mechanisms are all horizontal changes. It does not occur laterally from parent to offspring, or one portion of the lineage down its lineage in its evolutionary tree. It's occurring across the tree, across species, across phylum, and across domain, right? We're transferring our DNA horizontally with other organisms. Students should be able to understand the mechanisms for bacterial horizontal gene transfer. And students should be able to differentiate between horizontal and vertical gene transfer.